Hello GN311 students. Today we are going to be talking about gene regulation, looking at thorny dragons and the development of spikes. So these two guys wanted to make some discoveries. They both noticed some differences in each other. He doesn't have any spikes, whereas he does. So looking at this development pathway leading to the formation of spikes, we'll first look at examining a wild type. So in here there will be no mutations. I will be blocked by P, preventing K from being blocked, and E is still stimulated, leading to the formation of spikes. And then when we look at P strain having a mutation, it won't be functioning. So there's nothing to repress I, which will then repress K, making it unable to promote E, and no spikes will be made. If there was a mutation in I, without I, S and P are still going to remain the same in the developmental pathway. But without I being able to repress K, K can freely promote E, leading to spikes still being formed. So looking at a mutation in I and E, S and P are still going to remain the same, but without I to repress K, and no E being present, E will not be promoted and no spikes are going to be formed. And our last mutation that we're going to look at will be between S and K. So with S being mutated, P is not going to be promoted and I will not be repressed, which is going to let it actively promote K. But since K is also mutated, it can't promote E and spikes are not going to be formed. So the dragons still had a couple questions. They wondered, why is it that if they have the exact same genome sequence, they still end up getting differences in their skin? This comes down to RNA splicing. So RNA splicing is a hypothesis for why not all cells in the skin produce spikes, despite having the same genome sequences. mRNA is spliced after transcription to code for proteins to be produced, but this process varies, allowing for the expression of different proteins to be produced. In this illustration you can see, this starts with the same genome sequence, yet two different proteins are formed. Thank you for watching.